Finding the research questions is probably one of the most important elements that a scientist or a physician scientist can have. And that really question tells you for the rest of your life. In 1973, we asked the following question. What is the driving force that produces prevention and makes a wide parity produce prevention? And the concept was known for many years. Also for 1700 already have been demonstrated that women that have pregnancy were protected against breast cancer. And then that was confirmed in 1960 by the work done in the United States and also abroad, and many other hundred publications, until we come with the idea that probably the reason why prevention is produced by an early first pregnancy because the mammary gland is modified. But in order to study this concept, that the differentiation of the mammary gland has something to do with the prevention induced by pregnancy, you need to study. And then we decided to go through hundreds of breast tissue of women of different ages and reproductive history. And we found that one of the first elements that you find in the young breast at puberty is these terminal ductal structures. And these terminal ductal structures and puberty start sprouting, budding, forming the one that we call alveolar bud. And this alveolar bud forms the first lobular structure that is called lobulus type 1. And the lobulus type 1, or terminal ductal lobular unit, is where cancer starts. Then, when successive menstrual cycle, the lobulus type 1 developing a more complex structure that is called lobulus type 2, and at least as the woman gets pregnant, the breasts remain like that. In pregnancy, they develop the lobulus type 3, and later on lobulus type 4, that is the lactating breast. Meaning that what we see here is that the breast at birth and even during puberty is a very undifferentiated organ, and only complete the differentiation with pregnancy and lactation. And when we separate all this data and try to put, in order to make sense between nulliparous and parous women, what we found? That nulliparous women have only lobulus 1 and lobulus type 2, but rarely they have lobulus type 3 and 4. And when they go to the older ages or menopause, the lobulus type 2 disappear and the lobulus 1 start appearing again. Instead, in the parous women, we found the whole array, lobulus 1, 2, 3, and 4, and after pregnancy and lactation, after the 4th decade of life, they start reducing, and the only structure that you start seeing is the lobulus type 1. Therefore, there was a very important concept here, that the differentiation of the breast is only obtained during the process of pregnancy and lactation. And why, when you see in the postmenopausal women that most of them look exactly the same, but the postmenopausal women of parous are protected where the nulliparous are not. Therefore, then we propose the following hypothesis. The reason why parity produces protection is because during the process of differentiation induced by this hormonal event, the genomic signature, the genomic structure of the mammary gland is completely changed. And this change in the molecular structure makes that the mammary gland of parous women are less susceptible to develop breast cancer. The hypothesis was very good. However, it took several years until we can demonstrate that. And took the consortium that we developed between our institution, the Fox Chase Cancer Center, New York University, and Umeå University in Sweden, with the support of the Avon Foundation. It was a project of $5 million project in order to do this. And we interviewed, due to the policy in the health system of Sweden, that every woman can go for a mammography every 2 years, healthy women in order to detect breast cancer. In that way, we interview more than almost 700 women, and from them we separate in premenopausal and postmenopausal women, and we only collected breast tissue from those women that really fulfill all the requirements that we want. At the end, we have 109 nulliparous premenopausal women and 113 postmenopausal women. And then we made biopsy of these women, and the biopsy was a 4-core biopsy that was taken from different areas of the breast tissue. 1 core biopsy that was not the spirotom, was another one, and we made the histological evaluation of the breast in order to see what kind of a structure we found there. And the other 3 were used for genomic sequencing. And what we found? The first thing that we found is that all the part of women clustered together, and the nulliparous women clustered in the other side. All the nulliparous women are the green one, and the parous one are the red one, meaning that are all the genes in this group of women that contain a specific genomic composition that is different to the nulliparous women. That in itself was something that never has been demonstrated. Parous women have a genomic signature that is completely different than nulliparous one, as we already have predicted many years ago. But the situation was a little more complicated than that, because when we started looking at different ages, 5, 10, 15, 20 years after the first pregnancy, what we found? We found that we are 3 types of genes. Genes that were constant from the post-pregnancy until the postmenopausal ages. Other group of genes that pick up after 5 years post-pregnancy and later on disappear. Another gene that will appear after pregnancy but later on increase at postmenopausal age. Now, this was very important. Why? Because the peak of genes that really appear after pregnancy in the premenopausal women and later go down were all genes related to the immune response. And interestingly enough, those immune response genes can explain why certain women develop cancer even with an early pregnancy, because their immune system is completely different from one group over the other one. But this immune response disappears after 5 years. The important ones are those ones that are constant. They're ones that really show up. And those ones are related to differentiation, development, and chromatin remodeling. Meaning that this work that has been summarized in 3 papers published in the literature demonstrated one important conclusion, that pregnancy, early pregnancy, induces a specific genomic signature that lasts from premenopausal to postmenopausal women. Now, what this genomic signature means? Genomic signature is like kind of fingerprint that is developed after pregnancy and comprise the, I will say, the expression of different genes. And those genes can be divided in clusters according to their process of functionality. And we found that cell differentiation was one of them, the other one related to the shifting of the stem cell progenitor in the mammary gland, and the splicing mechanisms. I will not talk about these 3 mechanisms because these mechanisms take at least 1 hour to discuss, and I only have 20 minutes. Therefore, the 2 major clusters of genes that I would like to discuss is those ones related to chromatin remodeling and long non-coding RNA. Now, what chromatin remodeling means? When you see a section under the microscope, the first thing that you see is the ductal structure, the lumen over there, and then you see cells surrounding the lumen. And then you see the nucleus, you find here that the nulliparous women have a very clear nucleus that is called euchromatin, because really the chromatin is very loose. But when you see the parous women, all of them have the same morphology in the lobular structure, but the nucleus is more dense, it's heterochromatin. meaning that the chromatin that you find in the parous women are condensed. Here in gray is the DNA that is coiling in certain other molecule called histones. Therefore, the chromatin is compact, meaning that it's not open to be read in order to express the message. In the next slide, it's more clear that. You see here all these DNA coiling in the histones, and when they are coiled together, they are forming a compact heterochromatin in which the gene is inactive. But when the DNA open and the histones are open, then the DNA is open to be read. Now, the element that influences the process of histone modification around the DNA is the methylation of the histone 3 specific lysine 27. And that is a very important mark in order to determine when the chromatin is silenced. In more detail, you have the chromosome there, you see the string of DNA coiling the histone, and the histone can be inactivated by methylation, phosphorylation, or activated by acetylation. I don't want to go in detail on that. The important is that the three mechanisms are the ones that produce compaction of the chromatin that make the chromatin inactive, and when these elements are not taking place, the chromatin is open and you can read the message. Now, we found that the nulliparous women don't have the lysine 27, and instead the parous women have a very strong reaction for that, indicating that that observation were together. Now, hold this information for a while. The other element that we found is long non-coding RNA. Long non-coding are different to the microRNA. The microRNA is around 20 base pairs, where the long non-coding are 200 base pairs. At the beginning, they believed that they were junk DNA. That was DNA that was there that nobody knows, that was evolutionary concern, that was not functional at all. The matter of fact, Watson, they said, this is junk DNA. It's not DNA that needs to be studied. But in general, the situation is not like we see in certain moment in history. They demonstrated that long non-coding regions have a very important regulatory process. They are regulating the way that the genes are transcribed. And then we said, what happened with nulliparity and parity? And we found 42 genes that are differentially expressed. 42 genes that very few of them have been described before in the literature. And 21 of them are activated in the parous and 21 inactivated in the parous and vice versa. And one of those long non-coding regions is the XIST. What the XIT is a long non-coding region that has the function to inactivate the chromosome X. Now, when this long non-coding region interact with certain other proteins like are called the polycomb, immediately a set of reaction takes place and inactivate the chromosome X, making the chromosome X in the woman one inactivated, meaning that from the 2 chromosomes in the women, only one is active. The other one is inactivated. It's like some kind of a sheet is covering the chromosome, making them inactive. Now, the problem is that in the process of cancer, in the cell transformation, this inactivation is lost. And the XIT is not present anymore, and the X is not inactivated. And you see here in red over there, the XIT present in the control cell, and when the cell is transformed, they disappear. And that is the one that has been called in the literature as a Barr body. If you take a smear of any women in the room, everybody will have a Barr body. But interestingly enough, when the cancer process takes place, you lost the Barr body, meaning that you are lost the XIT. You are losing that. And in men that have breast cancer, this inactivation is more frequent. Okay, meaning that XIT has something to do with chromatin remodeling. Now, how this works? The XIT, such as I said, interact with certain group of protein that is called the polycomb, and that is the one that they produce inactivation of the chromatin. And you say, what this has something to do with the process of parity protection? The thing that is going on is during the process of pregnancy protection, the XIT is elevated and is very highly activated. And that is extremely consistent. We found this observation when we are doing our study 10 years ago, and we didn't find any explanation. We didn't know what was the meaning. What was the meaning? What is the meaning of this? Until recently, when we made RNA sequencing and made the XIT start coming back again. And then we found that XIT bind one of these proteins, this polycomb, and together with the lysine 27, the histone lysine 27, form the compaction of the chromatin. Now, this is good. Means that we have a very good idea why the parity produces protection, because the chromatin remodel in a different way than the nulliparous women. The problem is not only to find that information. This is a very good academic interest. It's how to control that, how we can manipulate this in order to produce prevention. And we decide to go back to our own model. Model is to take rat, to give the carcinogen, and we know that when you give this rat carcinogen, 100% of the animals develop tumors. Therefore, how we can use that? Then the thing that we have done is to make the animal pregnant. The pregnancy in the rat lasts 21 days. When you make pregnant, you wait, give the carcinogen, and what happens? All the animals that get pregnant before the carcinogen get 100, almost 95% protection. Meaning that we have now a model in animals that can mimic the human situation. And when we studied that, what we found? We found exactly the same thing. We found that the mammary gland differentiate, that the same gene that were activated in the women are also activated in the process. But then was not enough, because pregnancy is good. We explain why pregnancy produces protection. We have the human data, we have the experimental data. How we can mimic pregnancy without pregnancy. And then we decided to start treatment the animal with contraceptive agent, norethine methranol, for example, or medroxyprogesterone. And then we decided also to use ACG, because ACG is one of the hormones of pregnancy. And we use different doses. And what we found? First, I would like to, we treat the animal for 21 days like pregnancy, and then we wait and give the carcinogen, and we measure the tumor response. But before going to this data, I would like to emphasize that the ACG that we are using is the placental hormone. Is the ACG that forms a dimer of alpha and beta, and that is produced by the in situ trophoblast. Please don't get confused with the hyperglossylated ACG that is produced in the cytotrophoblast and is associated with malignant process, like the one that you see in molydatiform or in choriocarcinoma, meaning that the only hormone that we are talking from the physiological point of view is the one produced by the in situ trophoblast, that urinary and recombinant ACG has the same structure. And also don't confuse this with the free beta ACG produced by malignant cells that don't have anything to do with the ACG produced by the placenta, and also don't have anything to do with the pituitary ACG that is produced in a very small amount. Okay, making those quotations, what are the results? This is in order to show you how the ACG look like at the molecular level. We have two units, alpha and beta, and they are joined to the receptor. And the receptor is a common receptor for many other hormones like LEH. And when we treat this animal with contraceptive agent with ACG, what we found? We found that the ACG, given 100 international units for 21 days, protect the animal for the whole life of the animal against breast cancer, meaning that we can now mimic pregnancy without pregnancy using this hormone. And this hormone was more efficient than any of the other hormones that you can treat or can imitate, like contraceptive or even the combination of estrogen and progesterone. And interestingly enough, we found that if you measure the number of intraductal proliferation of carcinoma in situ that are perineoplastic lesion, ACG reduced significantly, meaning that ACG not only produced the total amount of tumor, but also produced the perineoplastic lesion, the precursor, are completely inhibited. Okay. But I am not going to too much excited because these are all data that are published in the literature. And we found, going back to our idea, that when we see the mammary gland of animal treated with hCG or pregnant, also the same phenomena exists. Methylation, the histone methylation, histone 3 methylated level lysine 27 is highly activated. Now, very good. I think that we found in human, in rat, whatever. But that's not enough for us. We need to have a system that we can control the mechanism in order to understand how the molecular mechanism takes place. And for that, what we have done, we take normal breast epithelial cell that is called NCSTNF, that is a cell that I developed in 1990. And it's a cell obtained from a woman, a premenopausal woman, 36-year-old woman, and we put in culture, and they get spontaneously immortalized, but maintain the normal karyotype. Now, when we treat these cells, that is the most normal cell that you can get in this moment in history, when you treat this cell with different doses of hCG, what we found? An increase in XIT, in the dose response matter. And also, when you see under the fluorescent, the intensity of the XIT is significantly increased when sequence versus the control. Meaning that we are able, in vitro, to induce the same phenomenon that we observe in the women and in the animals in vitro. We can see the effect. And the most important part is that you treat the cell for 24 hours or 48 hours, and the effect persists 5-10 days in culture. That the 5 days in culture is like 20-30 years. Meaning that this is a very important concept, indicating that something permanent was induced by the hCG at the level of the gene. Now, that was not enough. If the hCG really has something to do, we also found that the treatment induced upregulation of the BRCA1 gene wild type, and also induced upregulation of the p53 wild type. And that explained the previous result that we have that we didn't know how to understand 15 years ago, that animal treated with hCG have increasing in the p53. And if you increase the p53, the normal p53, or the normal BRCA1, what you will expect? That the cells repair the damage more efficiently, and also the cell can go from the high proliferation to low proliferation and programmed cell death. Meaning that we found here is that the hCG was able to control the activation of the XIT, BRCA1, the wild type, and also p53, the wild type. And also we observed that was accompanied by differentiation. And that this phenomena persists after the treatment for 5-10 days after. Now, in order to demonstrate that this was related to repair, we take the cell, we irradiate it with gamma radiation, and we measure one marker. That in this case is the histone H2HX, and we found that when radiation exists, increase. But when we put hCG, decrease, meaning that the repair takes place. We can see under our own eyes how the process of repair is taking place by the action of this hormone. Meaning that what we found here is that also, in addition to the activation of XIT, BRCA1, p53, and increasing the repair, was also methylation of the same histone in lysine 27 that we found in human. In summary, I don't want to enter in more complexity of this. What we found is a very interesting and unique pathway. hCG activates XIT, a low non-coding region that this process, the activation of this process, downregulates a series of genes that are also downregulated in the part of women. And the downregulation of these genes produce upregulation of the wild BRCA1 and the wild p53. And all of them, together with the methylation process taken by the histone, produce chromatin condensation and produce that the cells are more differentiation and able to repair better. This is a new paradigm. The fact that you can change the chromatin structure of a cell, making a susceptible cell refractory to carcinogenesis. And also that this is the mechanism that made a part of women protected and an only part of women no. And also demonstrate that you can do that by the single injection of hCG in the animal or in putting the hCG in the cells. But again, this is not enough. And then is when we demonstrate finally that what happened with pregnancy can be induced by the hCG. Now, then is when the Belgian group plays an important role. In a meeting similar to that, in 2014, I present the idea that maybe we can do a clinical trial. And Jack Janssen and Herman de Pijper can say, okay, I think that we can do this here. And we say, okay, great. We need 23 women that are carrier of a mutated gene because they're the only one that the ethical committee will approve. And we need to treat them for 3 months, but they need to have 3 biopsies. One at 0 time, other 3 months, and after 36 weeks from the beginning. And they say, yes, I think that we can do that. And they did it. And now we are analyzing those molecular structures in order to determine if with the hCG in these women are producing the same mechanism that we described before. Dr. de Pijper will describe what he found clinically. Unfortunately, only in July of next year I will have all the data, but everything indicates that we are in the right direction. Therefore, the take-home message is the following. I don't know what happened with this, my last slide. I don't know what happened. I cannot. Okay, I cannot make my last slide. Okay, that's it. Okay, the take-home message is the following. We found that recombinant hCG can produce chromatin remodeling, and that there are 2 important components, the XIT gene and the histone methylation as a marker. And that is the one that produces the cascade of events, producing DNA repair, and also blood cell transformation and differentiation. This suggests that we might be able to reprogram mammary gland cell by recombining hCG treatment to reduce the cancer rate. I mean that we are talking here some kind of one treatment, one series of doses, reprogram the chromatin of the cell and make this cell different. That is a novel concept. And that is what we are looking for. And the other important concept is that we can generate this by recombining hCG, that we don't need pregnancy anymore. And I would like to finally thank I don't know what happened, okay, this one. First, Jack Janssen for his collaboration, and first, all the women of the Limburg district that have been really extremely generous to donate their tissue in order that we can start this study. And also Herman de Pijper and Eric de Jong that have been instrumental in the collection of the tissue. And of course, the people in my group that have been able to obtain this information. Thank you very much.